Mr. Nuraj has more than 10 years of experience in handling biology for senior secondary students. In the years 2016 and 2017 he received the Best Teacher Award for his innovative teaching methodology.
Today I have one question, and please go through this question, okay? Go through the question, go through each and every option, and you can find the answer. And once, whether if your answer is right or wrong, whatever, I will explain to you. Insertion and adding an extension, that's the correct answer, and I will explain what happening there, okay? So that's the correct answer. And now you see, this PCR, it's a method which is widely used in the molecular biology. Okay, it is for the rapidly to make many copies, that means millions to billions copies of the specific DNA sample. Okay, and how it is done? It is done by, suppose can see here a genomic DNA. That means the DNA which of our choice. Okay, the DNA which and we have taken a small part of that particular target sequence, and now this target sequence, it will be heated at around 94 to 95 degrees Celsius, which is known as the first step of this PCR, that is denaturation. When it heats, what happens is the two-stranded or double-stranded DNA that will be separated into single strands.